All right, hi everybody. Let's get started for today. Just one final reminder that paper discussion number 3 is on Monday. So we spent a lot of time talking about the chemical senses so far. We talked about olfaction for a week or so, and then a couple of lectures on taste. And so today we're going to wrap up chemosensation with a discussion of a kind of third system for identifying chemicals, which is the vomeronasal system, and its role in identifying specifically pheromones and chiromones. So just as a introduction, some definitions. I'm sure everybody intuitively kind of understands what a pheromone is, but there's a little bit of a tweak to that definition in case you're not aware of it, which is that it's a chemical signal released by an animal that triggers a response in another animal of the same species. Okay? And those responses can be anything from fear or aggression to social recognition or mating. The sort of salient defining feature being that it's recognized by a member of the same species. On the other hand, a chiromone is a chemical signal that's released by an animal that then is identified by a member of a different species. Okay, and one of the key features here is that chiromones benefit the recipient. Okay, so these are things, for example, like this little hamster. If this were a real picture, which I'm sure it's not, but if it were, would be recognizing, for example, the odors, chiromones, coming from the cat, and this is eliciting fear. Any questions about those definitions? So this is a cross-section of a rodent head. And we've talked already about the main olfactory epithelium, or MOE here, which projects to the main olfactory bulb, and then where neurons project to various places in the brain, including, for example, the piriform cortex. So that's what we talked about already, and that's the main olfactory system. The vomeronasal system is separate. It involves the vomeronasal organ, which is actually present in this kind of bony capsule between the nasal cavity and the mouth. And projections from the VNO go to an area called the accessory olfactory bulb, or AOB. And from there, project to the vomeronasal amygdala. We've talked a couple of times about how the amygdala is kind of involved in, innate emotional responses. It also actually mediates some learning, but this is sort of the classic role for the amygdala, and so it would make sense that something like a pheromone or a chiromone would go through that area. So you might be wondering, how then would an animal actually detect chemicals within this kind of bony capsule? And there are actually openings in many animals up here above the teeth, and they can expose that area in something called a Flayman's response, which I'm sure you recognize in this horse, right? When they're doing that funny thing where they lift up their lips and make that sound, they're actually not just sort of doing it for fun. They are taking in volatile chemicals and into the passage to the vomeronasal organ. The molecular organization of the vomeronasal organ bears some similarities to the main olfactory system, except that the working parts are all different. Okay, so the receptors involved are just like ORs, GPCRs. Okay, they are from two families called the V1Rs and the V2Rs. And although they are GPCRs, they are unrelated evolutionarily to the odorant receptors. If you look at the neurons within the vomeronasal organ, you see that one receptor is expressed in each cell of the vomeronasal organ, which I hope sounds similar to what we've talked about in olfaction. However, unlike olfaction, where every neuron expressing the same receptor converges on a single glomerulus, in the vomeronasal system, neurons expressing the same receptor actually target a number of small glomeruli within the accessory olfactory bulb. Okay, or AOB here. There's actually two zones of the accessory olfactory bulb. The anterior zone is the target of neurons expressing V1Rs, and the posterior zone is the target of neurons expressing V2Rs. So there's a learning objective here to describe the organization of the mammalian vomeronasal organ. Are there any questions so far? Okay, downstream of the V1Rs and the V2Rs, there is a classic G protein coupled cascade that the details of which you should not worry about, but there's one thing that I do want you to remember, and that is that both V1R and V2R signaling require a common signal transduction channel downstream called TRPC2. Okay, it's labeled as TRP2 here, but the new nomenclature in the TRP family designates this as TRPC2. And this is required for VNO function. Okay, it's required for both V1R and V2R signal transduction responses, and so without it, you essentially have a non-functional VNO. This should bear some similarity to you to, like, for example, TRPM5 in taste, right? Which is required for sweet, umami, and bitter responses. This is the same sort of thing. We've got a TRP channel that is required for multiple receptor types signaling pathways. So people have used this little fact here that TRPC2 is required for the VNO function to molecularly essentially ablate the VNO and see what happens to a mouse's behavior. So this is a video of a wild-type male mouse that is encountering another mouse that's been castrated and then swabbed with male urine. Okay, you may ask, why would you do that to a mouse? The reason, as far as I understand, is so that the castrated mouse does not fight back, and you can just measure sort of the behavioral response of the other mouse. But as we'll see, the urine plays an important part in the recognition, and so the castrated mouse doesn't have normal urine, so you swab normal male urine onto the mouse and see what happens. And as mice are prone to do, they're not very nice animals, and they just try to fight. Okay? So the male mouse is essentially just attacking the castrated male with the urine, and they just do this. On the other hand, if you have a male mouse in the same situation, but that male mouse is mutant for TRPC2, the behavior is very different. okay? This may seem like a subtle difference, but actually this mouse is trying to mate with the other male mouse, right? And so this is kind of like the opposite response. Instead of trying to fight the mouse, he's trying to mate with him. So this is obviously a huge phenotype, right? A big switch in the mouse's behavior, and it's quantified here. So in these graphs here, the minus group is the TRPC2 mutant, right? This is wild type here, and this is a heterozygous control. So basically, they have one functional copy of TRPC2, so they should behave largely like wild type. So first thing you can notice, if we just look at wild type, if you look at the average attack frequency and compare the situation where there's no urine painted on the castrated male, which is in white here, versus urine painted on the castrated male, they attack the mouse much more when there's urine on the target mouse. Okay, this is presumably because the urine contains pheromones that elicit this response in the attacking male. We'll get into those pheromones in much more detail later. For now, just remember that urine is important. If you quantify the attack duration, you see the same thing. And of course, in both cases, the mutant mice basically never attack the target mouse. On the other hand, if you measure mounting frequency, then the wild type mice never mount the target mouse, but the mutant mice spend basically all their time mounting the target mouse. Okay, so it's a complete sort of binary switch in the male mouse's or the mutant or wild type mouse's behavior. So this led the researchers of this paper, which actually is fairly old now, came out in the early 2000s, to come up with a model, which is shown here. And so the basic idea of this model is, if we look over here first, that if a mouse encounters another mouse, its default behavior is to try to mate with it. Okay, and so that's illustrated here, where mating is kind of always activated by other sensory cues, whereas aggression is inhibited. Okay, typically like arrows mean activation, and then these like, I don't know what the proper term for that is, but the little line at the end of the arrow means inhibition. Okay, so that's kind of the default state. And so the model is that a TRP2 mutant mouse, or TRPC2 mutant mouse, that lacks VNO cues just sort of defaults to this default state. On the other hand, a wild-type male mouse receives cues in the VNO which lead to sex discrimination. This was a while ago. People were a little bit less cognizant of the difference between sex and gender. So I'll just say that this is sex discriminate or sex identification. Sex and gender are different, of course. Mice do not have a gender because gender is a social construct relating to someone's own self-identity, whereas this is talking about biological sex, right? So the VNO cues lead to the identification of the sex of the other mouse, and through the activity of V1Rs and V2Rs can lead to the identification of that mouse being male, and then that will suppress both of these default pathways. So it suppresses the mating, and then it suppresses the suppression of aggression. Okay, and if you suppress the suppression of something, you're actually activating it. So that's the model. And I should say that this is a very kind of hand-wavy model. Doesn't involve any real mechanistic underpinnings, right? Aside from the fact that the VNO is required for this behavior. So a lot about this model could be wrong, but it's kind of the framework that they set up based on this observation that mice lacking pheromone detection through the VNO had this switch in behavior. Any questions about that set of experiments? Yeah.
Why is it that even in the knockout mice, like when you add the male pheromones, there's like an increase in them wanting to spend time?
Yeah, great question. Why, if they're lacking VNO, why do we have this increase, right? Which they don't show whether that's significant or not statistically, but I think that it looks like it probably might be. Certainly it's trending in that direction, right? And so that actually reveals a little bit of a complication in the field in general that people tend to sort of sweep under the rug, which is that the main olfactory bulb plays a big role in this stuff as well. Okay, so they are probably smelling components of the urine through the main olfactory bulb, and that is enhancing something, whether it's sort of increasing arousal that then just leads to increased mounting, or, you know, you see, well, you don't see it there because they're mutant, right? But it's doing something that aids the recognition somehow. Maybe it's telling them where the mouse is or whatever, yeah. Any other questions? Okay. So we have a learning objective here to describe the importance of pheromones to mating and aggression in mice based on these experiments. So what could be the identity of the pheromones involved in this? Okay, so we've seen that urine seems to be important. And it turns out that later, the same people that did that study that we just talked about looked at the components of urine and identified a family of proteins called the major urinary proteins. Okay, these are abbreviated MUPs. And if you look at these MUP genes, you see that mice have a lot of them. They have 20 of them. In the brackets, I think, are pseudogenes. I think so. Yeah, I think it's basically the number of functional genes, and then in brackets, the number of pseudogenes, which, remember, are genes that exist in the genome still, but are non-functional because they've evolved mutations that render them non-functional in some way. Okay, so mice have a lot of these genes. Rats have a bunch, but a little bit fewer. And then humans don't have any. Okay, and other animals have few as well. At the same time, they looked at the number of genes that encode V2Rs in these different genomes, and they saw something striking, which is that the animals that happen to have a lot of MUPs also happen to have a lot of V2Rs. Okay, and so you can see that in the mice and the rats here. And so this led to the very shaky idea that maybe MUPs are some sort of signal that mice use, and then this signal is received via V2Rs. Okay, because they seem to have co-evolved, at least in number, in these various species. So there have been a number of studies on the role of MUPs, including at least one on the use of MUPs in social identification between mice, so pheromones, okay? We're not going to talk about that today. What I am going to tell you about is a story involving MUPs and their use as chiromones. So one of the really wild things about these innate signals is that certain animals are born to have stereotyped, hardwired responses to specific cues that are actually really important for them in the natural world. For example, if you put a mouse that has never experienced anything other than, you know, its mother essentially, and maybe its sibling mice, and I guess human handlers potentially, but it's grown essentially in captivity and isolation, and you just put odor in the cage from a rat, which is a predator of the mice, a cat, or a snake, they're all predators of mice, the mouse will show a fear response. Okay, so they are innately fearsome of these animals. So they show these behaviors called risk assessment, where essentially you see this mouse is kind of like checking out what's going on at the other side of the cage here. Mice are actually typically very interested in novel objects, but you can see that this one is kind of a little bit scared, right? He's having all these risk assessment episodes where he's obviously hesitant to approach that object because he detects these odors from the cat. So this is quantified here. This is just an image of the cage, right? They've divided it into area 2 and area 1. Area 1 has either a control cotton swab in it, so it's basically just the cotton, or a cotton swab that has been rubbed on the face of a cat. Okay, cats have lots of pheromones, you know, on their face. That's why they rub it on things all the time. And then they're quantifying the amount of time that they spend in the area with the swab versus the other area. And then they're graphing it in a super strange way that nobody understands when they first look at this, for good reason. I have literally never seen anybody else graph something like this in my life. But regardless, what they decided to do was compare the control versus cat situation and measure the amount of time they spend in area 1. So basically what this is saying is that when there is a cat odor present in area 1, they spend less time in area 1 than if there's a control odor present, right? And then they also do it for area 2. And what they're saying is here, and this is the part that tricks people all the time, when there is a cat odor present in area 1, the amount of time that they're spending in area 2 goes up, okay? So the odor is always in area 1, but then they're graphing the amount of time spent in area 1 versus area 2. Any normal person would graph the amount of time spent beside each other, area 1 versus area 2, for like cat versus control, but they did it the opposite. I don't know why. Anyway, you can see that a normal mouse, if you compare like the control area 1 to control area 2 here, typically spends their time investigating that novel object, whereas the odor causes them to not investigate it, or it does these risk assessment episodes. If you then quantify the risk assessment episodes here, you can see that they are very high in the presence of the odor and not so high without the cat odor present. If you take a mutant for TRPC2, it's the same thing as TRP2, right, that we talked about earlier, and you measure their risk assessment episodes, you can see that they go way down, basically down to baseline, right? And this is true both for rat odor, snake odor, and then as a control, They also look at the effect on wild type from rabbit odors, and rabbits are obviously not a predator of mice, and they find that there is no effect on risk assessment episodes by the rabbit swabbing. Okay, behaviorally, any questions so far? All right. So how is this mediated? The next question they wondered whether this is mediated by the VNO. It seems like it probably should be because these TRPC2 mutants lack their responses. However, getting back to what I said earlier about the main olfactory bulb, one of the related things that people in pheromones don't like to think about is the fact that actually TRPC2 does have a function in the main olfactory epithelium as well, just not as prominent a function as in the VNO. Okay, nevertheless, the next question was, is there activation of neurons within the VNO in response to these various odors? Okay, and to do this, you know, if I asked you, how would you do this? I think that the natural response based on what we've talked about already would be like do calcium imaging, right, in the VNO and see whether you puff on these odors and see whether it activates these neurons. This is a bit of an old school way of doing kind of the same thing, and it's using this gene called CFOS. You may have heard about CFOS before. This is an immediate early gene. It gets expressed at high levels when a neuron is very active. So the advantage of this is that it's much simpler to deal with, especially in regions of the brain that are hard to access with advanced imaging techniques, okay? So what you do basically is you take the mouse, you expose it to the odors, and then you sacrifice the mouse within 15 minutes or so. You do a section through the area of the brain or VNO or whatever it is that you're interested in, and then you stain for CFOS and you ask how many neurons are activated or are expressing CFOS, and this is a proxy for the number of neurons that are activated during that exposure to the odor. The big downside with this, of course, is the temporal resolution, right? You don't know for sure that these are activated, you know, in any time window smaller than like 15 minutes or so. So they could be activated in response to something else that occurs during that time window. But nevertheless, it's a lot easier, and so people are attracted to it. And what they see is that if they measure the percentage of cells within the VNO that's activated or expressing CFOS following exposure to various odors, you can see that snake, cat odors both elicit increased activation of the VNO. I guess rats and rabbits both do as well to a lower degree. Why rabbits are activating this? And, you know, I guess you would say they're activating non-fear VNO responses, but who knows? But these are all significant compared to the buffer alone. So this is making kind of like a time and place argument about the VNO's role in this response, right? The neurons are activated by these odors during exposure to them. They then did the same thing in the accessory olfactory bulb. And in this case, what they're actually doing is exposing the mice to a recombinant MUP. So why this is important is that this is a pure protein now, okay? So this is not urine. This is a MUP where they've taken the gene that encodes that MUP, they've expressed it in bacteria or something like that to produce the MUP, then they purify that protein, and so they're adding pure protein and seeing whether it activates neurons in the accessory olfactory bulb. So this really, points towards the role of MUPS per se, and not like some other component of the urine in these responses. And obviously, they see a big activation of neurons within the accessory olfactory bulb following exposure to this recombinant MUP. And so this led to the idea that these MUPs are not only involved in interspecies communication, potentially in aggression between mice, for example, recognition of a mouse's siblings is something that these MUPs have been postulated as doing versus intruder mice. But then they've also evolved to be these signals, these chiromones, that signal the presence of specific predators. Okay, and it turns out that these predator responses, the chiromone responses shown in orange here, are context independent. So essentially, just the presence of those MUPs elicit a behavioral response, as we've seen, right? So just the MUP on a cotton swab will cause fear. If you put a mouse MUP or mouse urine on a cotton swab, the mouse will not attack that cotton swab, though. Okay? So what they've said then is that the aggressive response, sort of the intra-species aggressive response, is context dependent. They kind of have to see that there's actually a mouse there to attack for them to elicit this aggression behavior. Okay, whereas the fear behavior is elicited in the absence of any other cues. Any questions there? All right. So far, we've basically talked about bad stuff, right? Fear, aggression, but not all pheromones and chiromones are used for negative things, right? Identifying other members of your own species or even different species might be used for more positive things. And so another study came out several years later looking at the role that MUPs may play as attractant pheromones between male and female mice. Okay, and so it turns out that female mice are attracted to recombinant male MUPs. Okay, so this is those MUPs here, right? And if you look at the preference index of female mice, you can see that they prefer going towards the MUPs versus the control. But here's the key and quite interesting thing here, which is that this occurs only when the female mice are in estrus. Okay, so only when they're fertile are they actually attracted to these MUPs. All right, as a reminder, this is the estrus cycle, right? Diestrus, proestrus, estrus here. So everything that's encoded in magenta in this figure is basically representing the estrus phase. And so if you compare this to the response of female mice in diestrus, when they're not fertile, those mice are not attracted to the male MUPs. Similarly, if you look at the responses in the VNO, you see much greater responses in the VNO of female mice who are in estrus. Okay, so that's quantified in this graph here, where the purple bar represents the response of female mice in estrus, whereas the gray represents the response of female mice in diestrus. So many more neurons within the VNO of these mice get activated when they're in estrus. So that's kind of interesting, right? And what it suggests is that this behavior is actually controlled not somewhere high in the brain where, there's some conscious perception of, internal state like estrus versus non-estrus and the chemical cues in the environment, but actually at the level of the sensory neurons themselves, which are responding more in this particular phase of the female mouse's cycle. Okay, so this I think involves like estrogen receptors and things. So there's cycling of hormones that actually directly activate or directly modulate the sensitivity of these neurons. Any questions about that? Okay, so as rodents go, mice, as I mentioned, are kind of mean. They're not really nice animals to deal with, and they do awful things to even their own young sometimes. They'll like eat them and stuff. So it's pretty bad, but not all rodents are so bad. So there's an example, one of the rare examples in nature of a monogamous mammal is this animal called the prairie vole. You've probably heard about them before, potentially. And these are socially monogamous rodents. They live in North America, pretty much across North America in various grasslands. And what happens is that in these prairie voles' lives, they at some point form a pair bond with a member of the opposite sex, and then they essentially co-occupy their nest with that other individual and raise their young with that other individual, etc. And these pair bonds actually remain intact, often for the life of the animal, and even after one of them dies, the remaining partner often will never actually mate with another vole. Very romantic, right? So, so these guys are a great example or a good model for studying kind of the social interactions between members of the same species. The bad part about prairie voles is that although people are actually working them up as a genetic model system in various places, it's much harder to do experiments on prairie voles than it is on mice. Okay, and so this is an old experiment, and it's a very kind of gross experiment in the sense that it involves surgery rather than like a nice molecular kind of change. But basically what this is showing is that if you measure the amount of time that a prairie vole spends with its partner versus a stranger, they will tend to prefer to spend time with their partner. Not all their time, but at least most of their time with their partner, right? And this is true also if you do a sham lesion, which basically means that they do a surgery, but they leave the VNO intact. But if you do a VNO lesion, then they no longer prefer to spend time with their partner. Okay, so this shows that the VNO is involved in either the formation of the pair bond. I guess I say here that it disrupts pair bond formation, but in reality it disrupts the pair bond itself, right? So expression or formation of the pair bond. Any questions there? Yeah.
Was it a significant increase in the time spent with a stranger or a decrease in the amount spent with the partner? Because it doesn't look like that much of a decrease of the time spent with their partner, but moreover, more time spent with a stranger.
Yeah, that's an interesting question. Yeah, because it's not like the total doesn't have to be 100%, right? They can just spend time alone, I guess. So, yeah, those VNO lesioned animals seem like they're spending less time alone and more time just with a random stranger. It's a good point. I don't know. I don't think they addressed that issue in the paper. And I don't know what the stats would be on all of that in the comparisons, but yeah, it's definitely a reasonable point. Any other questions? So the last thing I want to talk about is the question of whether humans have pheromones. So if we look at this evidence from kind of what we know about pheromone detection in other animals, there are a bunch of strikes against it, okay? So even though if you look at a embryonic human during development, they start to develop something that looks like a VNO, but then it gets lost later on during development, okay? So we basically have no structural VNO. We don't have anything that appears to be an accessory olfactory bulb. The TRP2 gene or TRPC2 gene is non-functional in humans, okay? It's a pseudogene. And V1Rs and V2Rs are also pseudogenes in humans. So none of the stuff that we've talked about being involved in pheromone detection or chiromone detection in rodents exists in humans. However, getting back to this point that was brought up earlier and once subsequently, it's possible that pheromones go through the main olfactory epithelium as well, right? We have intact odor detection in the main olfactory epithelium, so in theory, we could be detecting things that qualify as pheromones through our main olfactory system. And certainly some companies would want you to believe that and actually sell pheromone-containing colognes, for example, that men can spray on themselves in an attempt to change the response, let's say, of other people in their environment. Okay, so that might sound crazy, but actually there's a study about this, and specifically looking at one component of male sweat called androstenedione, or AND. And this is a derivative of testosterone. It is, in psychophysical tests, given a wide variety of descriptors, anything from putrid to woody or floral. So different people have different percepts of this chemical. But a study was done basically looking at the various parameters of women's psychological states before and after the exposure to androstenedione. Okay, and so what they did was they measured the change in positive mood here, and in when exposed to just control odors versus androstenedione, it seemed like everybody got into a worse mood during the experiment, but that worsening of the mood was less in the presence of androstenedione. The change in the reported sexual arousal actually was higher in the presence of and not only that, not only these like reported metrics, but also the absolute levels of cortisol in the circulation went up in response to the presence of androstenedione. Okay, so this is having a physiological and behavioral or perceptual effect on these subjects. Okay, so maybe there is some evidence that pheromones do something. So the last study I'm going to talk about, I'm mostly just talking about for fun. It is obviously full of caveats that, don't necessarily implicate pheromones, but, could in theory. So this is a paper from a number of years ago where basically the experiment was that these researchers went into a strip club and essentially asked the dancers to report their earnings and tips over the course of their menstrual cycle, okay? And what they saw was that the dancers actually, the normally cycling dancers, right, who are not on birth control, hormonal birth control, the normally cycling dancers actually earned more tips during their fertile phase than during the other phases. Okay, obviously this can have a lot to do with a lot of stuff, right? Whether it's their own mood or behavior is different during these times, but in theory it's possible that they also are releasing some sort of, cycling pheromone that affects the men's behavior as well. Relating back to this study that we showed before, right, about the behavior of female mice in estrus in response to male pheromones. I'll also say that this is worth looking at just for kind of amusement. If you have ever wondered what it would be like to hear a scientist describe in a surprising amount of detail exactly what goes on at a strip club, I recommend reading this. It's actually pretty funny. All right, any questions about anything today? Yes. I didn't check. Is that true? Is that what it says on the syllabus? That sounds right. I don't want to confirm or deny now, but that sounds right, yes.